Good morning for everyone. Thank you for this opportunity for being here and your attention. My name is Thatiane. I'm going to present you HIV-1 production. It's dependent on the incorporation of dynamine 2 by NEF. This work was performed starting UCSF and then developed at University of Brasilia. And I'm a professor from Instituto Federal from Brasilia, and I've been working on those both institutions in Brazil as full professors. Just a little bit of remembering about HIV-1 virus particle and other lentivirus virus particle, what is incorporated on them. We have here all the structure of the virus particle, the envelope, and inside the capsid, the two RNAs, but what is interesting is what comes inside this virus particle. We know about protease, integrase, transcriptase, reverse, also some accessory proteins like as NEF, but we also have some proteins of the cell like as clathrin and dynamine. So we are going to basically discuss a little bit from dynamine, which is a very important protein that is associated for cell exostase and releasing of the emerging vesicles. So about HIV genomic organization, we have here some accessory proteins, accessory genes that codifies proteins like as NEF. NEF, the negative factor, is on the late gene sequence here, as you see. And also you have really important proteins like as structures as GAG, and also enzymes proteins that are from POL gene. About the virus can be produced only by expression of GAG proteins, well known in the literature, just by have a vector which express the GAG gene, you can produce virus-like particles. But GAG and GAG-POL, they share the same mRNA for producing the structured proteins, and when the mRNA go forward for the expression of the POL region, it is produced the protease, transcriptase, reverse, and integrase proteins, which are really important for the virus replication. About vir protease is responsible for the cleavage of all those structured proteins for producing the virus particle. All this cleavage here are really required for being performed to produce a really infectious virus particle. And any not really cleavage of any of those sequence here abrogate or reduce really the viral infectivity. Despite of that, we have also some other proteins that are not structural enzymatic, but are really important for the virus pathogenesis like as NEF. NEF, it's really been discussed, and sometimes like not really essential protein because it's an accessory, but its importance is about how it increases the virus pathogenesis and can help us to elucidate a little bit of the virus pathogens of HIV and SIV. NEF, so, is a crucial factor for viral infectivity and has about 27 to 35 kilodaltons. NEF has been characterized to downmodulate MHC1, so avoiding the recognition of other CTL cells, also has been described to alter synapse of TCR signaling, also has a role on downmodulation CD28, downmodulation CD4, also interfere on the virus transcription by altering LTR activity, and on the virus production has really few results from NEF function. And the last paper was published last year by our group that had a result that NEF interacts with protease, and this can interfere on virus protein incorporation. But this paper here is going to go forward. Not only some virus protein can be interfered by the presence or absence of NEF, but also dynamine was before published to be a really keyhole for HIV pathogenesis since it interacts to NAF. And this group here, from Gottlieb group, they had a really important role on the literature of NAF by HIV pathogenesis when they describe about the interaction with NAF and dynamine 2. So we have other dynamines depending on the cell type, but dynamine 2 is the one which specifically interacts with NAF. And they have shown here that when you have the mutation of NAF, which abrogates the driving of NAF to the cell surface, you have here a phenotype of delta NAF virus. So here in blue, we are always going to check here about this previous paper published, which we have the Y-type virus, the virus with NAF mutation, and a virus with no NAF or delta NAF virus, which when we have absence of NAF, always we have reduction of virus infectivity, which this paper brings us, and was really important, was the whole taken about NAF when we have dynamine being expressed. So in this result here, this group showed by the transfection of a vector which expressed dynamine, the overexpression of dynamine plus virus production, you have a release of virus which was really full effective. But if you use a dominant negative dynamine which has a mutation that abrogates its function of dynamine, you have reduction of effectivity of virus, Y-type virus from HIV. On the other hand, when they compared the overexpression of dynamine or dynamine mutant on the context of delta NAF virus, you didn't have so evidence of reduce of impairment of activity by dynamine or dynamine mutant. On the other hand, they also tried to use sRNA to knock down dynamine, and in the absence of dynamine, it's observed that HIV-1 reduced its infectivity. So based on this result, our group wanted to challenge and to observe how NAF could contribute to viral infectivity by ligating, by binding to dynamine 2. So our objective was to investigate the effects of interaction between dynamine 2 and NAF during HIV-1 production. For developing this work, we use plasmid of PNL 4.3 plus antibodies against HIV-1 proteins and also dynamine and clathrin proteins. We also use plasmid for expression of dynamine fused with GFP. Our experiments were using cell-produced that produce for production of HIV, like as HEC293T cells and infectivity cells for SACE, like as TZMBL, and also some RNA interference sequence that was gotten by literature. So for virus production, we did the transient transfection into 93T cells, incubating lipofectamine with the plasmid vectors, and the supernatant was used or for analyzing the virus production or for doing infectivity assays. What was different from other works, what we did here, which is kind of pretty important compared to other published paper, that we characterize our virus doing iodixanol gradient. So usually our papers use sucrose gradient, that by fractions you can find that usually on the middle to late fractions you have the virus being sedimentating. So as you add the virus on the top of the gradient and then do the ultracentrifugation, you have a fraction that you can find mostly the virus particles that are infectious. What is really important of this methodology compared to sucrose, that As the virus is being produced by cells, you have also exosomes that are released from cells. And when you use the methodology of sucrose gradient, the exosomes can be found on all those fractions. So it has already published paper that shows that using udixanol gradient, while exosomes became retained still about third fraction, infectious virus are after that, so you can distinguish between really infection and mature virus part compared to those virus particles that are retained in exosomes fractions. So as a result, we have here the production of HIV Y-type virus and HIV Delta-9 virus by overexpression of dynamine 2 fused with GFP and dynamine 2 as control, which has a mutation, K44A. So based here on HIV-1, you have here all the over-level expression on the cells, but here I'm showing just the virus produced, the VLPs released from the supernatant and ultracentrifugal concentrate to show you here by ultracentrifugation. And all those viruses were quantified by ELISA P2N4, and the same amount of virus was used to infect a cell indicator of infectivity like STZ-MBL. And here we show how the overexpression of dynamine contribute for the infection or impair the infection. So as shown here, we have in the first lane the HIV-1 Y type, which was really important for our work that the first time shown that overexpressing dynamine, we had increased at least 2 times of the infectivity of HIV Y type, which previous work didn't show any impact of overexpression of dynamine. So considering that dynamine is really important for virus infectivity by NF, why didn't before no one shown that expressing more dynamine in the cell could really increase the virus infectivity as we shown here? And as control and as expected, use the dominant negative dynamine, you have the reduction of virus infectivity. On the other hand, on the context of Delta-9 virus, when we overexpress dynamine, we unfortunately couldn't increase or recover the infectivity of Delta-9 virus for the load of at least the Y-type virus, which really shows us that it's necessary to have NF to recovering this infectivity by dynamine. And on the other hand, we also try to show the results by using siRNA. And on this work, we use siRNA against dynamine, siRNA against clathrin, and both knockdown on the context of Y-type and Delta-9 virus, which was different from this work compared to PISAT that I was shown before, was the sequence that I got, the siRNA, which he got one sequence from his own published paper, and I got others from previous published paper that shown that was really great siRNA sequence. So we did the knockdown of clathrin and dynamine, and what I'm showing here for you is the virus production in the context of lane 1. All this here, Y-type virus, just clathrin knockdown, dynamine knockdown, both knockdown for HIV Y-type and Delta-9. We used the same amount of virus particle to infect cells, and when we saw the infectivity, we could see that infectivity of Y-type virus was decreased in the context of knockdown of clathrin, knockdown of dynamine, and both knockdown. We saw this reduction of infectivity in context of knockdown for HIV-1 Y-type and also for HIV-1 Delta-9. On the context of clathrin, the reduction of effectivity was shown before from Gottlieb group too, so this wasn't really new result. What we could here really emphasize is the knockdown of dynamine really reduced and confirmed previous work of effectivity of HIV-1 Y-type, and both knockdown abrogate even more the effectivity of HIV-1 Y-type, while those effective reduction is also seen in HIV-1 delta F, which previously wasn't shown. And how could really dynamine be impairing the effectivity for Y-type and delta F virus? So what's shown in the literature really that dynamine is incorporating virus particle, but the really absence of result is where's dynamine, where's the picture of the VLPs carrying on dynamine, which is really hard to do. So for developing a technique to really see the incorporation of dynamine, I had to use an overexpression system of dynamine, which we have the dynamine that is regularly expressed inside cells, but also overexpression with a plasmid vector. I used the supernatant, which we had the virus production in context of overexpression. And I used eudixanol gradient, which you can see here, Y-type and delta F virus being produced. And they are here mostly on fraction 5 to 9. When we analyze for the same gels, how was the context of dynamine? You can see here that dynamine is incorporated in both particles, both virus particles, but you have here really more dynamine on the fractions that are considered to be exosome fractions when compared to be really a mature virus particle in Y-type virus than delta F virus, which really emphasize that delta F virus, yes, incorporate dynamine. Which mechanism it incorporate dynamine, we have to really investigate. But the amount of dynamine being incorporated in this virus is really reduced compared to Y-type virus. So for doing this result, we really had to produce a really great amount of virus and concentrate them for then adding to those gradients, which before wasn't done. Other thing that is really hard for understanding the incorporation of dynamine is I use a lot of dynamine antibodies for N and C terminals, and has other works that have shown that dynamine 1, 2, and 3, they are spliced, and sometimes they can be just one of their isoforms, and those antibodies we are not really sure which isoform is incorporated of the virus particle. But using N and C terminal antibodies for dynamine, I could observe this incorporation. All those experiments were performed more than 3 times, and this here is just a representative of one of the assays. And also, how could we explain the difference of result of previous work from Gottgler group, which they use Zuckert cells, compared to mine that I use overexpression system, which is to 9-3 T cells. For answering this question, we was curious. We had the curiosity to check what is the dynamic expression level between those cells. So here you can have a representative amount of cells. There are like T cells, lineage like MOD4 and Zuckert, and also endothelial cells like HeLa, TZM, and 293 T cells, which we compare to beta-actin expression level, the expression level of the dynamine too. And here you can see a ratio between that. And we substantially saw that on Zuckert cells and MOD4 cells, which are T cell lineage, usually they have more dynamine available being expressed on the cells. This is not only the first work that is showing that, because other work shown that depend on the cell lineage, on the where the cells being produced, like brain compared to liver, lung, testes, you have different expression level of dynamines. And for explaining this difference of previous work to our work of the impact of dynamine on Y-type and delta F virus infectivity, we are showing here that in the context of more available dynamine too, like as MOD4 and Zuckert cells, you have more available dynamine for being incorporated on the virus particle. As you have more available dynamic to be incorporated in the virus particle, and dynamic being incorporated in the virus particle in the context of NAF, it's really important to increase the virus effectivity. That's probably the reason that those T-cell lines you have in the absence compared to the presence of NAF, about 4 times difference of virus effectivity. And when you go to the context of endothelial cells that you have less available dynamine to be incorporated in the virus particle, the presence of NAF is really more important to drive this dynamine to inside the virus particle, and also as you don't have so much of so much protein available for being incorporated, you can find effectivity difference between 2 to 4 folds, sometimes maximum 8 folds, when the virus particle is produced on those cells and logically analyzed on other effective indicator cells. So by this, I conclude that this data suggests that NAF and dynamine 2 interaction is really important factor for virus effectivity and production. I would like to acknowledge my supervisor in Brazil that I started to do my postdoc there 2 years ago, and nowadays I'm a professor there too, and also Boris Matijo Peterlin that I did part of my master and PhD in UCSF, all the support by CNPq, which support research in Brazil, and the institutions that I'm connected to. Thank you for your attention.